But what I've discovered over the last 10 years is that you cannot keep up with this information. What is in this book is about a year old. The information's about a year old. When you look in the literature, you will find something different. So this information is our understanding about a year ago. Does that make sense? And the information is going to change. What I want to emphasize, and what I want you to grasp, is the flow of things. There'll be some specific signals I want you to know, but I want you to get a feel for the flow of how things bind a receptor on the surface, and then things happen inside the cell. Okay? The specifics are going to differ between the signal, the receptor, and the different types of cells. Okay, I want you to get a feel for the flow. Okay, with that in mind, we have to talk about some receptors. The B-cell receptor, as you know, is a surface antibody, but there's a couple of other peptides that will bind that are essentially co-receptors. The T-cell receptor has the two peptides, the alpha and the beta. For most T-cells are the gamma and delta. And then there's the CD3 and the CD4 and or CD8. Okay? There's also cytokine receptors. There's receptors for a variety of different signals, but these are the ones that we're going to emphasize in here. Signals bind to those receptors, and then there's transduction. Transduction is what happens after the signal binds the receptor. It changes things inside the cell. Transduction's going from the signal and the receptor into the cytoplasm. Okay? And that's going to vary based upon the receptor. Okay? So we're going to see some variety, some variations here. There's usually several cytoplasmic signals that are generated. Some of them are enzymes that modify proteins. The net result of most of these receptors being bound by signals, the net result with most of them is a change in transcription. Genes are turned on and other genes may be turned off. So that what results as a consequence of these receptors being bound is that there's different proteins made within the cell. Different proteins are made in the cell. Okay? And that's because transcription factors are activated. Others may be turned off, but most of the processes we're going to discuss in this chapter involve transcription factors being activated. So you're going to have genes being turned on. Now, if you just look at, B-cell receptor and T-cell receptor, what happens when these receptors are bound by antigen? What happens in the big picture? The cell's activated, right? The B cell's activated when its surface antibody binds the antigen. It's activated. It starts dividing. It starts secreting antibodies. Does it make sense that you're going to have a change in the transcription? Yeah, you're going to have the genes involved in making that antibody turned on big time, so that those antibodies are secreted. And you're going to have other genes turned on that lead to cell division. So I hope that big picture makes sense. Okay, now we have to go through some details, unfortunately. The signal is a ligand that binds a receptor. Ligand is a general term for something that binds. We've talked about that in the lab. Oftentimes with cytokines, the receptor actually forms a dimer like this when the signal binds. This is called a dimerization receptor. But if you notice, in this is a relatively simple receptor. In this simple receptor, there's portions of the receptor proteins that are anchored in the membrane, but then they extend into the cytoplasm. Can y'all see that? And these have enzymatic activity. These are kinases. the cytoplasmic extension of the receptor protein has a kinase activity. A kinase is a protein that adds phosphate groups to a protein. A protein kinase adds phosphate groups to a protein. So, this is some biochemistry, but you have to cement in your head. Kinase adds phosphate groups. Those phosphate groups will in turn activate or deactivate the proteins to which they are bound. Now, that's something you discuss in biochemistry, but we have to deal with it in here. You discuss this in cell biology now?
Molecular.
In molecular, yeah, this is big in molecular. Okay, so protein kinases add a phosphate, and the protein to which it's bound is either activated or deactivated. In most of the cascades that we're going to talk about, that's an activation process. Adding the phosphate activates the next thing in the cascade. Okay. Now, protein phosphatases are enzymes that remove phosphate groups. So kinases add, phosphatases remove. Okay? So technically, phosphatases could also activate or deactivate proteins by removing that phosphate group. Does that make sense? So a kinase may activate, a phosphatase may deactivate. Okay? Or vice versa. Depends on the protein to which that phosphate's bound. Okay. So here, we see that when this receptor binds the ligand, it forms this dimer, and that activates this kinase activity. So proteins that come into contact with this enzyme may have phosphate groups added to it. And oftentimes when this kinase is activated, it has a phosphate bound to it. These little red dots are going to be phosphate groups. Does that make sense? Okay. You can see where this structure could activate other proteins by adding phosphates to them. Okay. In this class, we have kind of the same thing. A little bit different structure, but you have the activation with the dimerization. Okay, another thing I want to mention are these scaffolds. Scaffolds are not enzymes, but these are protein structures that allow multiple proteins to bind. Multiple proteins can bind this, and if you notice, this is a scaffold. It's not phosphorylated. A kinase binds it, and now it has these phosphate groups bound to it, and now other proteins can bind it. You see the sequence that can occur? So this can facilitate the activation of other things by activating one scaffold protein. But the scaffolding protein itself is not an enzyme. G proteins. Oh, a lot of proteins we're going to talk about are G proteins. These are proteins that bind GTP. And a GEF is a G exchange factor. I don't think we'll talk about it. RAS is an important enzyme. It's a small G protein. We'll talk about it. When you have this transduction occurring, you have amplification of the original signal. Sometimes, depending upon the cell and the type of receptor and the signal that binds the receptor, you may have a change in transcription if only one or two receptors are bound by that signal. With other signals, it may have to bind more receptors in order to get a change in transcription. Does that make sense? But with all of them, you have an amplification. You have the activation of maybe one G protein, and it in turn activates a number of other things, and those in turn activate a number of other things. You can have different enzymes different molecules being made, which bring in another signal one signal's activated, and that in turn brings in another signal, multiple copies of another signal. We're going to go through these examples, but the amplification means you may have just only a few, one or two or maybe a couple handfuls of signals binding receptors on the cell, but you have a large number of copies of signals made in the cytoplasm as a result. And that makes a drastic effect within the cell. Okay. Lipid rafts. The new edition of the book does not have this figure, but I included it. And it's on your handout, I hope, of your PowerPoint. Lipid rafts are structures you need to know about. It's areas primarily of the plasma membrane that have a high cholesterol content and usually a definitive composition of phospholipids. Don't worry about that. But it's an area on the plasma membrane where it's more solid. And, a membrane has the fluid mosaic structure. That's an old term, but the fluid mosaic where things can move laterally. And phospholipids can flip-flop, but proteins can never flip-flop. Most things have a lateral movement. Lipid rafts are little regions of the plasma membrane that have a high cholesterol concentration, and each lipid raft usually has its own collection of membrane proteins. And it's a more structured area, so they have the fluid mosaic. They are modal, but they are modal as a unit. They move as a raft. That's a great name. Those proteins usually have a function that's related. I'm going to give you an example here, which is going to make sense, but they have related functions, so it's important for them to stay together. Think TCR and CD4, right? They both bind the MHC. It's important for them to be together, right? They are thought to be maintained on lipid rafts together. When cells, we're going to talk about this later, but these lipid rafts oftentimes will, I'll talk about polarization later. I'll bring that term up later. I don't want to bring it up here. Cytoplasmic signals are made during transduction. After the ligand binds the receptor, then things happen in the cytoplasm. Cytoplasmic signals have a limited time of effectiveness. Proteins may be made, they may be activated, but they don't stay activated all the time. These protein signals that are made within the cytoplasm have a normal turnover. They don't stay around for a long time. Just like plasma cells. Plasma cells are formed during B cell activation, right? They're part of the cells, they're the largest group of cells in the clone. But plasma cells don't divide, right? Plasma cells don't divide. They stay around for a few weeks and then they die. So the proteins involved with cell division are no longer turned on in a plasma cell. Even though a few days earlier, when it was a B cell in the B cell clone, those genes were turned on. You don't want those genes, those gene products present all the time. You don't want them active when that cell becomes a plasma cell. Does that make sense? And so there's a normal turnover of those protein signals. And oftentimes ubiquitin is a protein that binds proteins that are going to be degraded. And if ubiquitin binds a protein, that protein's degraded in a proteasome. We've talked about that before. That's a cylindrical structure that proteins go in and they come out as peptides. It chops them up. But there's a normal turnover. And some of these enzymes and proteins that are activated there during this process don't stay active all the time, hours or days, and then they're destroyed. Antigen receptors are involved in activation of B cells and T cells, and I want to talk about the B cell receptor and the T cell receptor in more detail.